All right, well, I'd like to welcome everyone to the last Polyploidy webinar of 2021. And certainly not least, we have two really wonderful talks today. Up first is Dr. Na Wei. Na is an evolutionary ecologist and scientist at the Holden Arboretum, which is in northeastern Ohio outside of Cleveland. Na received her PhD in ecology and evolutionary biology from the University of Michigan. And following that, she moved to the University of Pittsburgh, where she did postdoctoral research with Teela Nashman and Aaron Liston from Oregon State University. During her postdoc, she focused on plant ecology, genomics, and wild strawberries. Last year, Na started her own lab at the Holden Arboretum, where she is studying eco-evolutionary adaptation in plants through the lens of ecology, genomics, and the microbiome. And today, Na is going to tell us about some of her research on the ecological consequences of polyploidy. With that, I'm going to let Na take it away.
All right. Thank you, Mike, for the introduction and for this opportunity to present our work. So to get started, I will briefly introduce some of my postdoc work as case studies to illustrate how polyploidy may confer ecological advantage. And then I will focus on some of my lab's current work on understanding how polyploidy may influence the species interactions. So polyploidy through genome doubling and emergence can have a profound ecological consequences. So for instance, it has been hypothesized that a diploid lineage that after polyploidy likely has higher fitness in changing environments relative to diploids. And one of such examples that came from wild strawberries, Fragaria. This genus has very widespread distribution across very diverse habitats. And it also has a very extensive ploidy variation, including diploids, hexaploids, octoploids, and even decaploids. And the genomes of those species have reviewed very complex and reticulate evolutionary histories between those diploids and the polyploids, indicating the repeated and independent events of polyploid speciation among taxa, and also within taxa among different populations. So by conducting a large-scale common garden experiment across 3 climatically distinct environments, the cool coast, temperate valley, and a very dry mountain region, we grew 269 genotypes from 72 different populations of 5 diploids and 6 polyploids. And in this experiment, we found the strong evidence for the polyploid fitness advantage consistently across the environments. And we further find out that the polyploid fitness advantage is conferred by both the functional trait mean and the trait plasticity by measuring a suite of leaf functional traits that capture essential plant ecophysiological processes. Those traits include, for instance, stomata science density that regulate plant CO2 intake and water transpiration, and specific leaf area that can influence the leaf carbon gain and leaf nitrogen content that supports photosynthetic potential, and the traits that are relevant to water transport, water loss, and water use efficiency. Those traits are leaf vein density, trichome density, and carbon isotope discrimination. So relative to the wild diploids and polyploids, the second example came from synthetic polyploids. So basically, the self-pollinated diploid seeds were treated with colchicine or water control. And this generated synthetic autotetraploid and 2 different types of diploids, that is the wild diploid and treated by unconverted diploid. And while the comparison between those 2 different types of diploids can inform a colchicine effect, and here we compared the treated by unconverted diploids with the synthetic tetraploid across multiple genetic families and species to assess how initial genome doubling would influence the functional traits and the fitness components. And in this study, we found that trait divergence occurs in the same direction and magnitude as seen in wild polyploids. And secondly, the fitness response to initial genome doubling is genetic context dependent, higher in some genetic lineages or genetic families, with the potential to fuel polyploid establishment and adaptive evolution. So these are two examples of the wild and synthetic Fragaria provide evidence that polyploidy can confer fitness advantage. But those two examples were evaluated in the absence of species interactions with conspecific or heterospecific competitors. So the question remains, how does polyploidy influence species interactions in response to environmental change? And here I want to emphasize that we focused on the species interactions at the same trophic level, that is the plant and the plant interactions, rather than species interactions at the different trophic levels, such as the plant herbivore interactions or plant pollinator interactions. And here we use the two case studies to evaluate the competitive interactions between the polyploids and the diploids. And these are two case studies that differ not only just in plant system, but also in the stress type. So in general, species interactions can be evaluated by examining both the intensity and the sign of the interactions. And here we focus on the relative interaction index, which the ranges between the -1 and the 1. And when it's above 0, it indicates a facilitation because the plant performance is higher with the neighbors than without neighbors. And then when it's below 0, it indicates competition. And the species interactions are often density dependent. So as the density of the intraspecific or interspecific neighbors increases, the intensity of a competition also increases and it becomes more negative. And for polyploidy, the competitive interactions that are expected to be asymmetric between the polyploids and diploids. And here the x-axis represents the interploidy density, that is the density of neighbors of a different ploidy level. So for diploids, the interploidy density represents the density of polyploid neighbors. So the slope reflects the competitive effect of polyploids on diploids. And here we just use the 4x as an example. And then for polyploids, the interploidy density represents the density of diploid neighbors, and the slope reflects the competitive effect of the diploids on polyploids. So now we focus on the slopes. The competitive asymmetry can be evaluated by comparing the slopes of interploidy competitions, that is, the competitive effect of polyploids on diploids expected to be stronger than the effect of diploids on polyploids. But the competitive interactions can be environmental dependent, and the stress gradient hypothesis provides a framework for predicting how the magnitude of interploidy competitions may change under stress. And specifically, this hypothesis predicts that competitive interactions should weaken as the environment becomes more stressful. And this is because individual competitors that are under stress are likely to impact each other less. But such the reduction in competitive effect is expected to differ between diploids and polyploids. So for instance, if the polyploids are more stress tolerant than diploids, they should be impacted less by stress. So their competitive effect on diploids should be reduced less than the effect of diploids on polyploids. So in addition to interploidy competitions, competitive asymmetry can be also evaluated by comparing the intra versus the interploidy competitions. So for instance, if the polyploids are stronger competitors, they should be limited more by individuals of their own, that is the intraploidy competition, than individuals of the diploids, that is the interploidy competition. And the opposite pattern is predicted for the diploids, that is the competitive effect of diploids on themselves is expected to be weaker than the effect of polyploids on diploids. And this pattern can be more pronounced under stress if the diploids are impacted more by stress than polyploids. So specifically for polyploids, the competitive effect of diploids on polyploids can be reduced more than the effect of polyploids on themselves. And likewise, for diploids, the competitive effect of diploids on themselves can be reduced more than the effect of polyploids on diploids. So to test those hypotheses, we focus on the diploid and the tetraploid Chrysanthemum indicum. And this species complex is native to Asia and has been introduced to other continents, including Europe, North America, and South America. And those are perennial plants that are widely distributed in China, especially the tetraploid, which has both the auto and the allopolyploidy origins. And seeds were collected from multiple sympatric populations of the diploids and the polyploids. And with those plants, we conducted a competition experiment where we changed the density of the diploids as shown from the left to the right. And we also changed the density of the polyploids as shown from the top to the bottom. And this 15 different density combinations form the basic unit of this experiment and were replicated 10 times with a total of 720 plants. And 5 replicas were under low water stress, where the soil moisture content was maintained at the 80% field capacity. And 5 replicas were under high water stress, which is drought, with only 20% of the field capacity. And we measured above ground the biomass to calculate the interaction intensity. And this allowed us to estimate the intraploidy and the interploidy competitions. So specifically in this study, we asked 3 questions. First, is there really competitive asymmetry between the polyploids and diploids in response to drought stress? And if so, does the competitive asymmetry translate into higher fitness in polyploids? And then lastly, how do functional traits contribute to the competitive dominance of polyploids? And for the first question, to test for competitive asymmetry, we first compared the interploidy competitions and our hypothesis showing on the left, and our results will be shown on the right. And for the interploidy competitions, we found that consistent with the hypothesis, the competitive effect of polyploids on diploids was stronger than the effect of diploids on polyploids. And then under high water stress, that's the drought, again, consistent with our hypothesis, the competitive effect of polyploids on diploids was reduced less than the effect of the diploids to the polyploids. And then in addition to interploidy competitions, to test for competitive asymmetry, we also compared the intra versus the interploidy competitions. And again, consistent with the hypothesis, for the polyploids, the competitive effect of polyploids on themselves was stronger than the effect of diploids on polyploids. And the opposite trend is found for the diploids. And again, as hypothesized, the competitive effect of diploids on themselves was weaker than the effect of the polyploids on diploids. And this pattern is predicted to be more pronounced under stress. And in our experiments, we found that in diploids consistent with the hypothesis, the competitive effect of diploids on themselves was reduced more than the effect of polyploids on diploids. But in the polyploids, different from the hypothesis, the competitive effect of the polyploids on themselves was reduced even more than the effect of diploids on polyploids. We suspect that this is likely due to the shift in resource use strategy of polyploids under the high stress, that such as the improved water use efficiency to alleviate the intraploidy competition for water under drought, which we'll talk about later. So for the first question, by examining the interploidy competitions and the intraploidy competitions, we found evidence for the competitive asymmetry between the polyploids and diploids in response to drought stress. And then for the second question, we ask, does the competitive asymmetry translate into higher fitness in polyploids? And to address this question, in the competition experiment, we accounted for all other factors that using the general linear mixed model and found that polyploids that had higher biomass than diploids consistently across the treatments. So for the second question, we found that the competitive asymmetry translates into higher fitness component in polyploids. And then for the last question, we ask, how do functional traits contribute to the competitive dominance of polyploids? And to address this question, we measure the functional traits in a separate experiment to avoid the potential influence of disruptive sampling on the competition experiment. And here we focus on gas exchange as shown on the top, including photosynthetic rate, stomata conductance, and the transpiration rate, and the water use efficiency on the bottom, including instantaneous water use efficiency during the photosynthesis and the long-term water use efficiency during the course of the experiment, and the integrated water use efficiency over the lifespan of a leaf based on carbon isotope composition. And in our study, we found that under low water stress, polyploids and diploids were similar in their water use efficiency, but polyploids that had a much higher gas exchange rate, which may contribute to their higher carbon gain and competitive dominance of polyploids under low water stress. And then under the high water stress, which is a drought, polyploids showed much higher water use efficiency while still be able to maintain a similar gas exchange as the diploids. So such improved water use efficiency may alleviate the intraploidy competition for water under drought, as we mentioned earlier, and that also may contribute to the competitive dominance of polyploids under the high stress. So for the last question, the higher gas exchange that under low water stress and improved water use efficiency under drought may contribute to the competitive dominance of polyploids. So now moving from drought stress to nanoparticle stress, here we focus on one diploid, Fragaria viridis, and one polyploid, virginiana, and ask, how do the interactions between the polyploids and diploids change in response to an emergent environmental threat, the nanoparticle stress? So nanomaterials or nanoparticles have revolutionized modern life, and they have been heavily used in all industries and our daily life, such as electronics, medicines, cosmetic, household, and agricultural products. But nanoparticles can be highly toxic to organisms due to their unique physiochemical properties. So for instance, they can damage cell membrane, interfere substance transportation, and they induce the production of reactive oxygen chemicals. So their intentional and unintentional release into the environment is imposing an emergent threat distinct from traditional stressors to the ecosystems and biodiversity. So two students from my lab are interested in understanding how nanoparticle stress may influence the species interactions. And to do this, they designed a minimal competition experiment. And the minimal competition experiment involved growing the diploid alone, or growing the diploid was the one intraploidy or interploidy neighbor, and the same for the polyploids. And the combination formed the basic unit of this experiment, and it was replicated 5 times under the control condition and 5 times under the copper nanoparticle stress. And this experiment was run for a total of the 12 weeks, but the data presented today only represented the first 4 weeks because we have not completed the sample processing. So different from the last case study we just mentioned, the minimal competition experiment here did not involve a series of change in neighbor density. So instead of the slopes, here we focus on the relative interaction index, the y-axis, and they compared the intra versus the interploidy interactions. And again, our hypothesis is showing on the left, and our results will be showing on the right. So for polyploid, we predict that the competitive effect of a polyploid on itself is stronger than the effect of diploid on polyploid. And in our experiments, we found that the result was consistent with the prediction. And then on the stress, we predict that the competition can be reduced according to the stress gradient hypothesis, and especially the effect of diploid on polyploid. And in our experiment, we found that consistent with the prediction, competition was reduced by moving up, and especially the effect of diploid on polyploid, which they become more neutral and even positive in many cases. And then for the diploid, we predict that the competitive effect of diploid on itself is weaker than the effect of polyploid on diploid. And then on the stress, again, we predict that the competition can be reduced, especially the competitive effect of the diploid on itself. And in our experiment, we found that different from the prediction, under the control condition, instead of competition, the diploid experienced a facilitation, benefiting from the presence of the intra or interploidy neighbor. We suspect this is likely due to the improved moisture retention with the presence of a neighbor when plants were still small with little roots. And then under nanoparticle stress, such a facilitation was reduced, becoming more neutral. This is likely because plants were smaller under stress, so were their ability to confer benefits. So in this minimal competition experiment, we found that the polyploid results support the competitive symmetry, that is, the effect of a polyploids on themselves is stronger than the effect of diploids on polyploids. But the diploid experience a facilitation, benefiting from the presence of the intraploidy or interploidy neighbor. And the nanoparticle stress reduced the interactions, that is a reduced competition for the polyploid and a reduced facilitation for the diploid. So overall, to summarize, the story of a chrysanthemum provide evidence for the competitive asymmetry between the polyploids and diploids in response to drought stress. And then the story of Fragaria further pointed out the complexity of interactions between the polyploids and diploids. And specifically, we found evidence for the competitive asymmetry in polyploids by facilitation in diploids. And nanoparticle stress affects both the magnitude and the sign of interactions. And then lastly, I would really like to highlight that all the people involved in those projects. I also want to thank you for your attention. I'm happy to take any questions if time allows.
All right, well, thank you, Na, for a really wonderful talk. Yeah, we have plenty of time, about 5 minutes for questions for anybody that has any. Go ahead and, yeah, folks can either type it in the chat or pop on their video and ask away. All right, I have a real quick question while we're waiting. I have a
Sure.
bunch of questions, but so just thinking about the nanoparticle stress. So how exactly does that happen? Are you putting nanoparticles in the soil, or are these applied to the plants, or yeah, how do you stress them with nanoparticles? I'm sort of curious just what that looks like in the greenhouse itself. Like what does that mean?
So in nature, nanoparticles could occur in both aquatic and like terrestrial system. So if in terrestrial system, typically in the soil. So that's the reason in this experiment we applied it directly to the soil. Even we apply around like 100 ppm, but eventually the nanoparticle will start to aggregate as that would occur in natural system. And for that experiment, that's actually part of a larger experiment to examine the interactions actually of the plant microbiome. So that's the reason it's actually occurred in the culture tubes.
Right.
Yeah, so the results I'm showing today actually in the sterile environment. So we have not completed the ending the microbiome into that. So I'm hoping next time we will be able to actually talk about that.
Yeah, that would be really wonderful. I look forward to that. All right, Jeanette has a question here. What do your results imply about the persistence of diploids? It seems like they can't win. And I was sort of thinking the same thing.
Yeah, so yes, in both the cases, we emphasize a lot about the polyploid advantage. I think that in our the first study I mentioned about like the wild strawberry across the 3 climatic distinct environments. In that study, we actually emphasized that a lot of times those could be like the special like segregation, both at the large and the scale, like the niche differentiation. So that it can allow the in terms of coexistence, they're not growing together. So the presence of both the diploids and the polyploids, but also could be related to dispersal limitation or like the demographic drifts or like the historical effect. All of those factors may contribute to the persistence of both the polyploids and diploids.
Yeah, I agree. Yeah. Let's see. Oh, another question here from Wilhelm asked, thank you for the nice talk. What effect could the increase in polyploidy induction from 2x to 4x have on the fitness results between diploids and polyploids? Could that explain the higher biomass?
Wait a second. So what's the effect of increased polyploid induction? Okay, on the fitness results of that higher band. Yeah, so that for the synthetic one, we find it quite mixed results. So basically, it could be like really high in some genetic lineages, but in some cases, so those that are higher fitness in the synthetic polyploids, it could even occur in the same population. I would say how to say that. So even in the same population, the genotypes may have really different results. So you can see actually it's not like the one population, all the synthetic polyploids that have the benefits. It's rather than quite mixed across the different populations. So that have the potential to feel like the multiple origins and polyploid establishment across the different populations.
All right. Oh, Brittany, it looked like you were going to ask a question. Is that right?
Yeah, I've got one here. Hi, Na, great talk.
Thank you.
I wonder if any of your work on these different, cytotype interactions looked across multiple polyploid cytotypes, because I know there's several papers at this point that show sort of a difference in niche partitioning between tetraploids and hexaploids. So I'm not sure I would expect all polyploids to behave the same way.
Yeah, so I totally agree. Actually, we indeed compared in the wild the polyploids across the 3 climatic distinct environments that the common garden experiment. We actually compared hexaploids, octoploids, and decaploids. What we found actually those when they become polyploids, the difference between them very small. But the biggest difference actually between the polyploids versus like diploids and polyploids.
Interesting.
Yeah, so yeah, we didn't, so for that experiment, so we didn't have like the tetraploid, but I'm expecting tetraploid will similar to other polyploids, but tetraploids that mostly occur in Asia, and we didn't have the permit to import them and grow them together.
Fair,
So,
yeah.
But look forward to see that results from our Chinese colleagues.
Cool, thanks.
Yeah, very cool. We have time for one last question here from Janita. Asks, the results of the study show that polyploids and like invasive plants help other plants to thrive under stress, correct? And then does this apply to both auto and allo polyploids?
Yes, yeah, that's the part a little bit challenging for us to figure out, so that because there are, for the strawberries mostly we have an allo polyploids, so we don't really can attribute that effect due to hybridization versus the genome duplication per se. But in our, so that's the reason we use the synthetic, the polyploids, but at the beginning, like initial genome doubling effect, it could be really stochastic in many cases, but we can hoping to see like the over generations of the effect. But also for the wild polyploids, sometimes a lot of times they have the both. Well, in our case, they have both the auto and the allo polyploid origins. So it's really hard to tease apart the effect. I did not address the question, but that's my feeling about the. It's a little bit challenging to attribute for the wild polyploids.
Yeah, it's a great question. I really look forward to seeing all the future work on this. It's and getting all these other variables in there too, as you guys finished up your work. It's really wonderful. Well, thank you, Na, for that great talk. And if anybody has any questions for Na, as they haven't had a chance to ask, we'll have a chance at the end to come back around and ask some more questions and have a little more discussion then. And so we'll switch over to our other speaker now, Dr. Alison Scott. I'll let Alison go ahead and throw up her slides. Alison writes that she appreciates polyploids big and small and is learning to love working in a new model system as a postdoc in the newly formed Department of Chromosome Biology at the Max Planck Institute for Plant Breeding. So with that, I'm going to let Alison take it away once she gets everything updated here, or up on the screen here. There we go.
Get all the right screens in order. Yeah. Desktop one. Share, and then, okay, do you guys see
we
my
see your
presenter
slides
view or my full slides? Okay, cool.
It looks
great.
good. Yep.
I'll move so I can see you guys. Great. Okay, cool. All right. Thanks for the introduction, Mike, and thanks so much for organizing this seminar. Thanks everybody for being here as well. So yeah, after a really long time of working on only intractable and large polyploids, a year ago, I started working on a new postdoc here in Polina Novikova's lab at MPIPZ in Cologne. And one of the systems that we work on is the Siberian Arabidopsis lyrata. So today I'll talk about some of our current work and some of what we found out about the genomics of adaptation to tetraploidy in this group. And so, there we go. When we study polyploids, and Na is a great lead into this, right? We tend to think about a few different aspects of polyploid evolution, right? And I'm framing these for now as polyploid emergence, so how polyploids originate, their establishment, and then their endurance. And largely, I think these can be summed up by a few different explanations. Some of you guys might disagree, but for emergence, I think it's largely agreed that there's either some genetic predisposition or there's response to environment or climate that contributes to the formation of a polyploid. When it comes to establishment, you can rely on something like asexual reproduction, or you have to adapt your meiosis in order to deal with the consequences of having all of a sudden way too many chromosomes. And when it comes to long-term polyploid endurance, it could be adaptive advantage or maybe just sheer luck. And I know plenty of ink has been spilled over the relative contributions of these two aspects. So to start, I'm going to talk a little bit about polyploid emergence and what these different components may be contributing there. this figure won't be new to anybody in this seminar, but I show it anyway. We know that polyploidy is more frequent at higher latitudes, which automatically gives us some indication that, okay, environment seems to have something to do with polyploidy formation. And when it comes to the genus Arabidopsis, we also know that many of the polyploids in this lineage arose during recent glacial maxima, which also says something about the contribution of environment, but which also can give some indication that this, we know this is a lineage that's predisposed to polyploidization. So it's a nice comparison spot here as well. And to look at both of these questions, we're looking at a lineage where we see a lot of polyploidy in Arabidopsis, but also adding this environmental component by studying ploidy variation in Siberian lyrata. So for those of you who aren't sure where Siberia is, like basically this whole gray area on this polyploid frequency distribution map, you can see there's a whole lot of missing information, and a whole lot of that is Siberia. So, it also happens that this fills in a little bit of the gaps there. On the right, top right, these are herbarium specimens, because for this particular part of the study, I'm just showing our herbarium samples. And then on the bottom, we have a distribution map where the colors indicate the ploidy level. So the red points are diploid individuals, the blue points are tetraploid. And then this is just a little neighbor joining tree kind of on the bottom right to look at the kind of clustering of these different ploidy level groups. And one observation here was that there's a handful of these tetraploids that tend to cluster together in the tree. They're also geographically clustered. And okay, this map doesn't look maybe like these things are this far apart, but this is like an entire continent, right? So these distances are actually quite large. And I remind myself that I don't work on conifers anymore. So like pollen is not traveling this far. Anyway, so this kind of led us to believe that there may be at least two different origins of polyploids in the Siberian lyrata, but maybe more. Okay, so just from this kind of initial study, we say, okay, ploidy does vary in Siberian lyrata, more tetraploids than we actually expected to see with multiple origins, but we still don't have any indication about the role of the environment here that might be triggering the formation of unreduced gametes, or whether meiosis is being impacted in terms of crossover numbers being regulated, or whether there's any adaptive advantage to polyploidy in this context. So I'll try to go through some of this. But first, how did we infer ploidy in these Siberian lyratas? So we did this in a few different ways. So the first was looking at the base frequency distributions using Inquire to look at the distributions only at biallelic sites, and then by this inferring a best fit to a different ploidy level, right? We also looked at the number of S alleles in each of these individuals, which can be quite variable. So this provides a good opportunity as well. And then we validated this with cytogenetics using live samples from some of the same regions that we have herbarium collections from. So I'll show here a combination of two of these data types. I'm showing you the overall picture first. This is work that was done by Uliana Kolesnikova during her bachelor's thesis in the group. So, okay, the color of each of these bars is the ploidy level as according to Inquire. So diploids are red, triploids are green, and tetraploids are this kind of teal, turquoise, blue, whatever you want to call it. The number on top of each of these bars is the number of S alleles that we found in each sample. And the size, the height of the bars is their heterozygosity. And these are just ordered from lowest to highest heterozygosity. This is not like, this has nothing to do with their geographic distribution. Okay, so one thing, the most exciting thing to me, first of all, was that it looks like Inquire is relatively reliable when it comes to assigning ploidy levels. So in the case of diploids, we consistently find, up to 2 alleles, but not more than that. And in the case of our putative tetraploids, we can find up to 4 different S alleles, which is like, okay, this is kind of compelling. It's always a little bit scary to take too many liberties with these analyses. But what we also noticed here was just that, okay, clearly there's something going on with these guys that have very low heterozygosity, but that our tetraploids are in general more heterozygous than the diploids, which supports the fact that they're actually tetraploids and that they're still outcrossing. Which is also interesting. Oh, this was just a zoom, so you can see these numbers better, but I guess we're kind of past that point. So in addition to these herbarium specimens, we've also been lucky to work with some of our collaborators who have done field collection of seed. So the points on the map here are, again, the herbarium vouchers, and then these rectangles are showing where more recent collections have been done of live samples and seed that we have growing in our greenhouse here at Cologne. And this is all thanks to largely Nikita and then Elena and also Uliana, who I mentioned, who has done some of these analyses as well. And you know, I've never done fieldwork in Siberia, but it turns out it's a much more dramatic and heterogeneous landscape than I expected to see. So you see not only some cute lyrata here, but some of the fauna that they were able to interact with, although it doesn't look near as easy as collecting redwood samples. You have to scramble up cliff sides and hike through fields. Although, you know, you do get to get around by helicopter. So the only way to get to some of these sampling sites is by, like, helicopter is literally the public transportation at some times of the year in some of these regions, which is cool. But then again, the mosquitoes far outnumber the people, the lyrata, anything else that you can find. But thanks to our intrepid field hands, we've got plenty of lyrata growing in now Cologne, which allowed us to grow some plants and then super zoom in and take a look at what's happening in terms of the chromosome counts. And so using this, we were able to validate both the inquire and the S allele assessments of some of these populations to say that we do indeed have tetraploid lyrata in Siberia, and then also diploids as well. So this is something helpful. We're working on getting some better images here, but are struggling a little bit with the growing conditions for some of these guys. So, okay, we addressed emergence. So we found these tetraploids and we know where they're growing, but then we of course are curious about their establishment and understanding how polyploids in Siberia and these lyrata are able to adapt. And it turns out that lyrata is a great system to study this because it allows us to look at parallel adaptations to polyploidy in different contexts. So there's the well-known case of tetraploid European lyrata that involves gene flow from Arenosa. There's the Siberian lyrata that I mentioned here. There's also a subgenome of lyrata that's part of the allotetraploid kamchatka. And then of course, we know from extensive work in Arabidopsis arenosa that these natural autopolyploids really are able to adapt their cellular machinery to whole genome duplication. So we know that from work that came out of Kristen Bonble's and Levi Yant's group over the years, that these natural tetraploids are restricted to only one crossover per chromosome, which it turns out is very helpful when making functional gametes. And when they tried to induce polyploidy using colchicine, they found that all of a sudden things became a mess during meiosis, which indicates that this is a process that took place over time, right? There's adaptation taking place. Furthermore, they also showed that the proteins that are under selection in this tetraploid arenosa are these key components of the synaptonemal complex. This is something that's also interesting for us in our department. Our department head, Raphael Mercier's group, studies this quite a bit. And another postdoc, Laia Capella Perez, just published this year in PNAS that the synaptonemal complex is imposing crossover interference in Arabidopsis. So there's some well-documented function here, and all of the Arabidopsis and polyploidy people in our sphere are very curious about this. So there's this one approach. We then know that from the work in Arenosa, then when looking at autopolyploid European lyrata, that there was introgression that helped to mediate this adaptation by borrowing alleles from Arenosa that facilitated the adaptation in European lyrata. Okay, so of course the question we had is whether there was any evidence for genetic adaptation in Siberian lyrata. And part of the reason we thought that this might be different is that we know that recombination rate can be impacted by temperature. We know that maybe it's possible that in these colder than usual regions, Maybe meiosis is happening without problems because crossovers could be limited anyway. So we looked to see whether there would be any evidence to support this. So this is just a quick and dirty, not quite selection scan to look at whether there are any genes here that are maybe under selection in the tetraploids compared to the diploids. So again, we weren't really expecting to see anything necessarily, but looking at the overrepresented Go terms in this collection of red dots above the line, which is what's interesting here, there were a couple that were related to meiosis, which is always interesting, especially in my wing of the department here. And then when I went to look precisely at what some of these loci were, we see again components of the synaptonemal complex that are under selection in these tetraploid accessions of lyrata. So in particular, ASY1 and ZIP1B. ZIP1B is the one that my colleagues just published a paper on recently, and some other genes that also tend to come up when looking at this in Arenosa and European lyrata. So ASY1, ZIP1, these here are two of these members of the synaptonemal complex. And so on this level of the work, at least, it seems that adaptation to polyploidy is converging on a genetic level because these components are under selection in both tetraploid Arenosa and Siberian lyrata. And I just want to point out that there is no Arenosa in Siberia. So there is no introgression from Siberia, or sorry, from lyrata, sorry, from Arenosa in Europe to Siberian lyrata. Like was the case in the European lyrata, we know that this is mediated by integration. This isn't possible in this case. So this was something very exciting and compelling, but of course there's lots more work to do. Something that we have ongoing now is further studying polyploid emergence in terms of how they originate. So we found these polyploids in Siberia, but we still don't really know how it is they got there. So in our live collections that we have outside and in the greenhouse and in growth chambers, we're looking at studying the frequency of unreduced gametes among all of our accessions to see if there are any populations that are producing more than we expect, or whether they produce more than we expect in general compared to European accessions. And then also growing these under different conditions to assess the relative impact of temperature on unreduced gamete formation. So I can summarize quickly, and then I have a little other story that I might quickly show because I wanted to make sure I have time. Okay, cool. So just a quick summary. For the emergence, we know that there are polyploid lyratas in Siberia. That's nice. In terms of establishment, we know that meiotic adaptation is taking place and that it's involving the same genes as in Arenosa. But what is to be determined and what I'm working on now is, for one, are the same alleles involved as in Arenosa? Are these guys just hanging around in standing variation in these other populations or not? If these polyploid populations are in fact of different origins, then what are those relative contributions happening differently? And of course, the relative role of environment is something that we're looking at as well. And sorry, I also should say that this sampling, so these samples, like these 2021 guys, they're just in the greenhouse. So we don't have any data from these yet either. So there's going to be more dense sampling also, both from the 2022 collection here that we just got some data back from, and then this most recent expedition from this last year. So we'll have more points to compare and denser sampling to cover some of these regions as well. It turns out it's hard to cover a lot of ground out there in Siberia. So let me skip to something else. Okay, well, I guess I'll thank these guys and then I'll go to some more. Okay, so of course, thanks to the Novakova group, our department at MPIPZ, especially the Mercier group, which has been great. And then Philip Kolar and Maida, we work closely together, and they, for example, have some of our seeds and are making synthetic polyploids from some of our Siberian seeds to see what comes out there. Okay, I'm gonna try for 5 minutes. Okay, so another question, I showed this kind of earlier to look at the different ways where we can look at polyploidy in the Arabidopsis clade, there's also an opportunity to look at the association between selfing and polyploidy. And so in terms of who are self-compatible, there's the two allotetraploid Arabidopsis species, Suessica and Kamchatka, are both self-compatible. And then we also have the diploid lyrata in North America is self-compatible. And I showed you before this figure where I highlighted on the far left, there are a couple of accessions that have very low heterozygosity compared to the others. And they have a 0 on top, which means that they have no S alleles. So this is something that we were curious about. And because we had seeds from these, we were able to grow them outside and bag them so that nobody else could pollinate them. Actually, this was still in the greenhouse, so it was even easier to control things, to make sure that they couldn't be pollinated by any other means, and found that they set viable seed. And so we said, okay, this is something interesting. So again, this was some work that Uliana piloted, again, as part of her bachelor's thesis. You like hear about students like this, but she's really incredible. And so we started by looking at the S locus, because in Arabidopsis, this is how self-compatibility, incompatibility is controlled. And there's these two components, the male component SCR, the female component SRK. Okay, we don't need all these animations because I don't have that much time. So basically SCR is on the pollen, SRK is on the stigma, and there has to be recognition in order to allow fertilization to take place. Otherwise it doesn't work. And there's a lot of divergence in these, or there's a lot of variation in these haplogroups, both at the SCR and the SRK locus. And so, okay, so looking at our Siberian lyrata, that was a sulfur, which is a diploid, by the way, to start. Okay, so this is a diploid, but I promise I'll talk about polyploids again in just a second. So we knew that it had this haplogroup S12, but the female component of the S locus was completely lost in our assembly. So okay, we found a sulfur with low heterozygosity. So of course, this is like a nice genome to assemble, right? So SRK is completely gone. The SCR that is there has lost the cysteine that's otherwise super conserved. So we think maybe it's not actually functional. So we look through, and from our other collections, we were able to identify, a few other sulfurs. So these turquoise guys are sulfurs, and then the red circles are outcrossers. So we found that all of the sulfurs have the same S haplotype, okay, at this SCR locus. They all, it's all broken in some way or another, right? But so this conserved cysteine is like missing multiple times. We also found a sulfur from the herbarium that has SRK, but not SCR, but that SRK is also broken. So all components of this are broken at different levels, which is really cool. And we're still working on this story. But where it also becomes really interesting is that lyrata is one of the parent genomes to allotetraploid Kamchatka. And Kamchatka is an allopolyploid that has multiple different origins. And when we look at our, let me skip this again. When we look at our accessions and we look at Kamchatka, it looks like our lyrata is more closely related to some Kamchatkas than those Kamchatkas are to other Kamchatkas, right? Potentially of these different polyploid origins. And so what we think happened here is that there was this loss of function mutation in one of the parents. Here we were proposing it's this lyrata, perhaps from our Siberian collections, that then promoted the establishment of allotetraploid Kamchatka, which is a sulfur, right? So it lost this self-incompatibility at some point and began to be able to self, which is obviously something useful when you're a polyploid. And because this is also something like what happened in Suessica and also recently published in Capsella bursipastoris, we think that this could be another pattern contributing here. So I don't have a summary for this because I wasn't sure I was going to show it, but There's some polyploids there, and I'm happy to take questions in the 5 minutes almost that I think I have left.
All right. Well, thank you, Alison. That was awesome. Yeah, and as usual, people should go ahead and post their questions in the chat box or pop on your audio, video, and ask away.
Yeah, and I don't think I can see the chat right now unless I stop.
All right, I'll let you know.
Okay.
All right. Well, I had a real quick question. Think about the S locus diversity. So you sort of showed, the number of alleles in each of the individuals. Do you have a sense of the sort of, the total number of alleles in the whole population, or I guess the structure of the S locus allele distribution?
Yeah, how the diversity is distributed among these. Yes. Right. Yeah, we do. I don't remember off the top of my head, but it's another figure as well, so we can see kind of who's pooled where. Yeah,
I was curious if that gave you any insight into the number of polyploid origins as well. If you haven't had a chance to look at that, but yeah.
Yeah, that's a good point too. Definitely. Yeah, thank you.
Cool. All right. It always takes
it's all good.
Everyone a minute to think about stuff.
Can I start a not quite formed question?
That's okay. I have those all the time.
So first off, great talk. Really found this bonus story interesting. So okay, I'm not super familiar with self-incompatibility in Arabidopsis. Is this a gametophytic system?
No.
Okay.
Sorry. So this is all happening. The interaction between SCR and SRK is happening between the pollen and the stigma. So it's like we've got a protein here. Yeah.
I'm familiar with I guess S loci recognition systems where if they are recognizing the pollen, then the pollen tube gets arrested. But it sounds like
the pollen tube won't grow.
Exactly.
Permitted. Right. And we've done some work with this too, like where we've got some fluorescence and everything too. And like the pollen tube can't grow if the SCR, SRK, whatever combination is not permitted. Yeah.
Huh.
Yeah.
That's I think potentially interesting, because it seems like what little research is out there on self-incompatibility in polyploids deals more with like gametophytic self-incompatibility systems where it's sort of the opposite mechanism where there's recognition and then arrest of pollen that's sort of related to you. And there's some work that sort of suggests a mechanism for how that's working, but in this I'm not sure that same mechanism would work. So do you have any idea why this one cysteine keeps falling out of genes? Or
no, we don't know. And again, this was like a surprise, right, to kind of see this anyway. So no, we're not sure. And with all these new accessions that we have, we're also checking in those cases to see what S alleles they have, if any, and whether that SCR locus is still intact, right, to see whether it's working or not. But yeah, like I showed on this map, it's happened multiple, if I can pull this back up, it's happened multiple times, it looks like, or there are multiple samples that are missing this SCR component. Okay, it's there, but it's not functional anymore. And most of them are completely missing this SRK. And again, like this map doesn't really represent how much space there is between, right, these accessions. So yeah, I'm not sure yet, but this is a good question.
Well, cool, thanks.
Yeah, thank you.
There's a talk there from Sylvia in the chat box. Says
yeah,
there you go. All right, I'll let you see that.
Wait, how do I think I have to stop sharing and able to actually.
No worries.
All right, done sharing. Wait, I'm still sharing. Stop share. Now here's all the people. Sylvia. Unreduced gamete formation will be estimating to pollen or only eggs. Oh, I mean, pollen to begin with. Okay, I love the idea of looking at unreduced gamete in eggs. I just don't know how feasible it would be to do that in a high throughput way. But if people have suggestions, I would love to hear that. Yeah, I don't know. And do we expect to see differences there in population accession species for unreduced gamete formation? I don't know. So we have found polyploids across Siberia. So it indicates that they're happening in different places. There is this one region that I showed that's kind of in the west. There's this peninsula where there were a whole bunch of polyploids. And when I first showed this herbarium specimen slide, it was like all these tetraploids that clustered together. And so maybe over here, there's going to be a higher propensity because none of the herbarium samples are diploid at all. So there could just be this kind of polyploid colony that's there. Although then maybe they're not really going to make unreduced gametes because maybe they're already all, if there's not diploids around, it's a little bit less meaningful. Yeah, it would be convenient to see differences in the populations, I'll say it that way, but I don't know if we will. Okay, and then a question I see, is there any reason why polyploids are better adapted to higher latitude than lower latitude? I think that's a great question. And I know there are some people here who probably have some thoughts on here too. I mean, I don't know that the argument is about being better adapted to higher latitude, and I certainly think, that there's a lot of evidence that it's just about occurrence happening more at higher latitude, right? So the more it's happening, the more likely that sometimes, whether by luck or by adaptive advantage, you're going to get established and you're going to persist there. So maybe, I don't know, I see a couple of knowing looks, and some people don't have cameras, but I can suspect that they might have something to contribute here too.
I go along with that for the most part.
Yeah, okay.
Yeah, for what it's worth on that whole topic, I think there's a lot to do with formation, right? And yeah, anyway. Yeah.
Less than adaptation, yeah, right.
Yeah. All right. Are there any other questions for either Alison or Na before we wrap up today's webinar? All right. Well, with that, I'd like to thank both Alison and Na for two great talks to close out the series this year. We'll be meeting again in the spring. I will post a sign-up sheet probably early next week with some dates. So for those of you that are interested in giving a talk, feel free to sign up when you see that. And if you're and you can shoot me an email too if you have a question about whether or not a talk is appropriate. And we're coming back around. So I posted a tweet last night. I was curious about this. We have, partly because it's getting difficult to email everyone, because my Outlook has the university's Outlook server fails when I try to send the email to everyone all at once, because we now have 825 people signed up, which exceeds the number of email addresses I'm allowed to send to at one time, which is astounding. So when we started this almost 2 years ago, I thought we'd have just a handful of people that would want to do this, and it's been really cool to see the huge number of people that have signed up and aspirationally interested in joining us. But the videos are posted on YouTube, and they're watched quite a bit, 100 times or so. And we've had almost, I think this is our 54 talks now, 55 talks. So it's been really wonderful, and I'm happy to keep pushing this forward and make it happen. So as usual, we'll have an open sign-up sheet. People can just sign up, and I'll have to poke some people maybe to fill it out the spring. But if you've given a talk already too, and you have some new stuff, we're hitting that point where it's fine to start cycling around. And I really enjoy this, and I kind of view this as like a great extension for my own sort of reading and lab meeting and everything else. And I'm happy to keep it focused on junior folks and just talking about all the cool stuff that's really happening. So in our community. And anyway, yeah. So please sign up, I guess, is the short version of this, and don't feel too stressed out about it or anxious about it. I say that and I give one of these talks and I would be super stressed, but anyway, hopefully no one else is. So, all right. Well, thank you. Yeah, I just see these. Thank you, Jeanette. And yeah, I hope everyone has a great holiday season and a happy new year. And yeah, we'll see everyone in 2022, which seems like it could be a good year for Polyploidy. I don't know. There's a 2-2 there, right? I don't know. We're gonna make some bad joke, but I want to thank everybody, and I really look forward to seeing how all this work goes. Yeah. All right.
Thanks, Mike.
Well, we'll see everyone.